The RTK pathway begins when the ligand epidermal growth factor, or EGF, binds to the extracellular EGF receptor, creating a dimer. This activates the receptor phosphorylation of tyrosine residues with ATP. RAF protein binds to the phosphates, activating the MAPK cascade through MKKK. This cascade phosphorylated MEK and ERK, turning them into MKK and MAPK. These targets then enter the nucleus to activate the transcription factor of CMYC that promotes cell growth and division. This triggers the start of DNA replication, where the initial parent strands of DNA run antiparallel to each other. Helicase then opens up the strands to begin replication. Two primates create the starting sequence for DNA replication, placing RNA primer along the parent strands in the 3' to 5' order. Two DNA polymerase continue from the primers and build daughter DNA strands that match the nucleotides of the parent strands. Building in the 3' to 5' direction results in the two DNA polymerase to go back and replace the primers with DNA fragments. Finally, two ligases are sent to connect all the DNA fragments to form two replicated daughter strands. Once DNA is replicated, cells in the interphase stage can go through the mitosis process. In prophase, chromatids condense into chromosomes and the centrioles move to the opposite ends of the cell while the nucleolus and nuclear membrane break down. In metaphase, the chromosomes align in the middle of the cell as spindle fibers from the centrioles attach to the kinetochores of the chromosomes. In anaphase, the chromosomes are pulled apart. The sister chromatids separate to the opposite sides, triggering telophase to occur where the chromosomes arrive at the opposite poles. As the spindle fibers and chromosomes disperse, the daughter cell's nuclei form. Mitosis ends with cytokinesis, where a cleavage furrow forms in the center of the dividing cell, resulting in two replicated daughter cells.